I'm happy indeed to introduce Roshan to you. Roshan studies how neuromodulators, in particular dopamine and serotonin, affect motivational and cognitive control. Over the years, she has made several seminal contributions, empirically and conceptually. For instance, she discovered and formalized mechanistically the impulsive side effects of dopaminergic drugs. She tackles such questions with broad experimental approaches, combining fMRI, PET, pharmacology, genetics, fine-grained behavioral analysis, and in particular computational methods to understand better the intricate interaction between specific neurocircuits, specific neuromodulators, and the behavior in the world around us. Roshan Kohls studied psychology in Groningen, obtained her PhD with Trevor Robbins at Cambridge, and did 2 postdocs, one with Mark Desposito at Berkeley and back at Cambridge. In 2007, she joined the Dundas Institute for Brain, Cognition and Behavior, where she leads one of the principal investigator groups, and she became professor of cognitive neuropsychiatry. Time is too short to mention all honors and awards she has already obtained in her still young career. Just to highlight a few, Roshan is member of the Royal Netherlands Academy of Arts and Sciences and the Academia Europaea. She obtained 2 Royal Society fellowships from the UK. She obtained the most prestigious awards for her career stage from the Dutch Organization for Scientific Research and the Royal Netherlands Academy of Arts and Sciences. She is member of the most relevant scientific advisory bodies for the Dutch government, and she is editor of both the Journal of Cognitive Neuroscience and the Journal of Neuroscience. Last but not least, Roshan is an excellent communicator, explaining terrifically well her research to the broader public and to the scientific community. Thus, I invite you to expect just the best when listening to Roshan Kohls talking about chemical neuromodulation of the adaptive mind. Please.
Wow, thank you, Guyen, for this wonderful introduction and for the invitation, of course. Great opportunity for me. Let me start by acknowledging my wonderful team and the fact that the work I'll present is the result of major collaborative efforts. I'll focus in particular on work done together with Martine van Schouwenberg and Hanneke den Ouden, and then move on to focus on work done together with Andrew Westbrook and Michael Frank in the context of teamwork, which was supported critically by Ruben van den Bos, Monja Vrobeuze, Lieke Hofmans, Jessica Mehta, Britt Lambrechts, and Danai Papatopetraki. Okay, arguably, we live in the age of the connectome, where the brain is conceptualized as a distributed computing system consisting of multiple structurally densely interconnected neural networks, which can now be characterized in exquisite detail. A key point for today is that these structural connections are relatively fixed. So a major question is, how does our brain adapt dynamically, flexibly, to the constant changes around us? And my take-home message for today is that the large ascending neuromodulatory systems originating from the midbrain, like dopamine, might be particularly well suited to fulfill this functionality. It is the chemical neuromodulators that adapt our brain to our changing environment. Indeed, the brain constantly faces a variety of computational dilemmas. Depending on task demands, our neural networks need to exhibit flexibility or stability. They need to exhibit controlled or automatic processing. They need to exhibit accuracy or speed, generalization, or multitasking. Now, for my talk today, I'll focus on the first of these 4 dilemmas, which I think are at the core of this fascinating psychological construct that is cognitive control. This construct is relatively ill-defined, but generally refers to that set of mental processes that allow us to obtain our goals by resisting distractions, temptations, and impulses. It's often considered a hallmark of the human brain, and accordingly, I have no doubt whatsoever that you have no problem to continue to focus exclusively on this lecture for the next 45 minutes without being distracted by any email or maybe phone messages, right? Or actually, will you really decide to make use of this exquisite capacity to focus? After all, focus is opportunity costly. Some of the new incoming input might turn out to be relevant. Some messages should be processed and require a form of cognitive flexibility. What we need is a meta-level ability to decide when to focus and when to let go of focus, when to rather exhibit flexibility, flexibly respond to the changes around us. How does our brain do that? Answering this question requires that we reconceptualize, I would argue, the problem of cognitive control as a problem of reinforcement learning and decision-making, rather than a problem of implementation per se. And my key take-home message that the chemical neuromodulators adapt our brain to its environment is in part grounded in the observation that they enable the arbitration or decision-making between different cognitive computational strategies. But let me start with a definition of chemical neuromodulation, highlighting a few key general principles of chemical neuromodulation. I will then zoom in on the dopamine system and also point to a major problem in psychiatry and neurology today, and that is that there is huge variability in both the direction and extent of dopaminergic drug effects, both across and within different individuals. Next, I will illustrate how the general principles of chemical neuromodulation can account for these paradoxical effects in the clinic, by taking you with me on a historical tour through previous pharmacological and neuroimaging studies, which really represent two separate lines of work, one focusing on striatal dopamine, its role in value-based choice and learning, and one focused on frontal dopamine and its role in cognitive control and working memory. Then, in the second part of my talk, I'll present ongoing work in which we begin to integrate these relatively segregated lines of work. And then I'll end with raising a few big open questions that arise from this work. Okay, so what do we mean by chemical neuromodulation? It generally refers to that set of chemical processes that curtail or prolong, augment or diminish effects of fast signaling in neuronal networks, often conditional on the baseline firing state of the postsynaptic cell. So this neuromodulatory mode commonly is commonly distinguished from the fast direct signaling of the classic neurotransmitter mode via glutamate and GABA. It is rather the control exerted on that classical neurotransmission. Now, by the way, before I proceed, I want to make the important point that, as is the case for most basic neuroscience, pretty much all advances in our understanding of human chemical neuromodulation have leveraged major insights from work with experimental animals. And indeed, the neuromodulatory systems are particularly well conserved across species, here shown on the rodent brain. Okay, now while neuromodulatory signals act, if you will, as broadcast signals due to their widespread innovation of large parts of the brain, their manipulation has radically different effects depending on where they act in the brain. So in the case of dopamine, for example, which innervates multiple relatively segregated frontostriatal circuits, we now know that the cognitive and motivational consequences of dopamine reflect modulation of dorsolateral and ventromedial frontostriatal circuits, respectively. And such regional specialization stems in part from differences between regions, for example, in the density of different receptor subtypes or receptor families. Also differences in the density of projection neurons, of the different projection neurons. And there are also differences across brain regions in the speed of reuptake. A second principle I want to highlight is the fact that these neuromodulatory systems exhibit exquisite capacity for self-regulation. So short latency, phasic responses are controlled by tonic levels of activity via presynaptic feedback inhibition of synthesis and release. And what follows is this well-established inverted U-shaped relationship, according to which drugs that increase receptor stimulation have positive effects on systems with low basal levels of activity, but paradoxical negative effects in systems with high basal levels of activity. So I've highlighted three general principles so far. First, neuromodulators have different effects by acting on different neural systems. Second, these neuromodulatory systems self-regulate to maintain equilibrium. And third, effects of neuromodulators depend on the baseline state of the system. I'd like to now zoom in on the dopamine system to illustrate these different principles. And the dopamine system is now well known to be implicated in a wide range of both healthy and disordered cognitive functions. The cognitive role of dopamine is also keenly illustrated by its implication, for example, in ADHD, but also Parkinson's disease, and even in healthy states of stress and fatigue. All these states and disorders are accompanied by impulse control disorders, cognitive control disorders, which can be remedied to some degree by dopamine-enhancing drugs. But here is our major problem. It turns out, unfortunately, that there is huge variability in these dopaminergic drug effects on cognition. Consider, for example, ADHD, where symptoms of inattention and impulsivity are commonly treated with dopamine-enhancing drugs like methylphenidate, Ritalin. But consider, on the other hand, Parkinson's disease, where dopaminergic medication can rather contribute to cognitive deficits. In some people, a considerable proportion of people actually inducing impulse control disorders, pathological gambling, for example. Indeed, we've shown that recently that pathological gambling addiction is accompanied by enhanced striatal dopamine synthesis capacity, as measured with dopamine PET. So how can the same neuromodulator have such different effects across different individuals? This is a key question that drives much of our research program. And this is not the only thing. Diametrically opposite effects are also seen within the same individual. This is nicely illustrated by a recent study by Sean Fallon, who gave people a small dose of methylphenidate and found anti-impulsive effects on a task of working memory, healthy volunteers, but pro-impulsive effects on a task of selective attention. So how can the same drug in the same people have such different effects across different tasks? Now let me take you through some of our previous studies to illustrate how we have begun to address these questions. As I said earlier, the dopamine community, until recently, was somewhat split, with some research lines focusing on striatal learning, value-based learning, and some research lines focusing on frontal control. Now, in the domain of striatal function, we've provided some early empirical foundations of the dopamine learning hypothesis by showing not only that there is a strong association between dopamine synthesis capacity, measured with PET, and human reward learning, but also between the effect of a dopamine drug on reward learning, plotted here on the y-axis, and individual differences in baseline levels of dopamine synthesis capacity, again measured with PET. So here you can see that a dopamine drug improved reward learning in low dopamine subjects while actually impairing, perhaps overdosing, subjects with already high levels of dopamine. So this demonstrated that the human dopamine system also expresses baseline dependency. And the immediate implication of this result is that in order to isolate dopamine's effects on cognition, we better take into account the baseline state of the system. In the domain of prefrontal function, we've contributed to revising the key hypothesis that the cognitive effects of dopamine reflect direct modulation of the prefrontal cortex. And instead, we've highlighted a role for striatal dopamine in prefrontal control. And the starting point of that line of work was the observation that cognitive control, working memory, are in fact multi-componential phenomena comprising not just the stable maintenance, but also the flexible updating of task-relevant representations in working memory. And based on various lines of evidence, not least the observation that there is neurochemical reciprocity between prefrontal dopamine and striatal dopamine, we put forward the hypothesis that dopamine might modulate the distinct, stable, and flexible aspects of control by modulating distinct prefrontal and striatal regions, respectively. To test this hypothesis, we developed a delayed response task to assess the separate stable and flexible aspects of control in terms of distractor resistance and task switching, respectively. And in line with this functional and neurochemical reciprocity, a series of studies with this and similar paradigms followed, showing opposite effects of dopamine on these distinct, stable, and flexible aspects of control. For example, in patients with Parkinson's disease who exhibit severe dopamine depletion in the striatum, we see increased task switch costs and decreased distractor costs, suggesting that cognitive flexibility is impaired, but cognitive stability is enhanced. Moreover, dopaminergic medication in these patients reduces switch costs, but increases distractor costs, biasing the system back to cognitive flexibility away from cognitive stability. Similar effects have been found in other populations. Subsequent pharmacological imaging work showed that a dopamine drug, in this case bromocriptine, often used to treat Parkinson's disease, modulates switch-related bold signal in the striatum, but not in the prefrontal cortex, whereas that same drug in the same people modulated distractor-related activity in the prefrontal cortex, but not in the striatum. So it's this kind of evidence that led us to conclude that dopaminergic drugs have contrasting effects on the distinct, flexible, and stable aspects of cognitive control by modulating the striatum and the prefrontal cortex, respectively. So the important point here is that control requires a dynamic adjustment of a balance between distinct brain systems, depending on task demands, and that such task-dependent adjustment might be subserved by dopamine. The next question we asked, but how exactly then does dopamine alter this balance between cognitive stability and flexibility? According to current gating models, striatal dopamine regulates the flexible gating of actions proportional to their costs and benefits. Critically, this role of striatal dopamine has been proposed to extend to the flexible gating of cognitive actions, cognitive tasks, and rules maintained in working memory. In essence, these novel models integrate classic top-down biasing models of cognitive control with classic models of the basal ganglia, where striatal dopamine lowers the threshold for gating. And to test these ideas, and inspired by computational modeling work from Randy O'Reilly's group, we set up a set of empirical studies to test the hypothesis that the biasing by the prefrontal cortex of task-relevant processing in posterior cortex is controlled by the striatum such that task-relevant processing in posterior cortex is flexibly updated only when striatal dopamine gates attentional switching. And in these studies, we used bidimensional face scene stimuli, where faces and scenes were overlapped. And on each trial, subjects had to choose between two of these bidimensional stimuli. And they did this based on either the faces or the scenes. And they were instructed to switch to the other dimension as soon as they detected that novel exemplars were introduced in this other dimension. Now, strong switch-related signal was observed in the striatum, as expected, as well as the thalamus and the frontal cortex. Conversely, strong attentional gain signals were observed in stimulus-specific sensory cortex, so that the FFA was more active when people switched to faces, whereas the PPA was more active when people switched to scenes. The full pattern of bold signal here was beautifully captured by a dynamic causal model in which striatal switch-related activity modulated the effective connection from the prefrontal cortex to this stimulus-specific sensory cortex. So this then suggested to us that striatal signal might gate the output of the prefrontal cortex. In a separate study, we also found that the dopamine drug bromocriptine modulated switch-related activity exactly in this striatum. And moreover, this region was right adjacent to a region exhibiting a strong link between a behavioral index of switching and white matter integrity, as indexed by fractional anisotropy measured with diffusion tensor imaging. Probabilistic diffusion tractography from this FA seed region in the striatum showed that the tracts originating in this basal ganglia region culminated in a region of prefrontal cortex that also exhibited a significant drug effect on functional connectivity with the striatum, here shown in blue. So what this together showed is that dopamine can modulate switch-related processing in a region of the striatum that connects structurally and communicates functionally with the prefrontal cortex. So what we conclude from this kind of work is that striatal dopamine potentiates cognitive flexibility by gating attention to a cognitive task rule via modulation of prefrontal control of sensory-specific posterior cortex. But the key question still remains: how then does striatal dopamine set the threshold for gating a cognitive task? And according to these gating models, and also currently popular resource allocation models, resources are allocated to a cognitive task so that they can be gated based on their expected value of cognitive work or of that cognitive task. And so this leads me to my current work, where we begin to integrate these dual roles of dopamine in value-based choice on the one hand and in cognitive control on the other. And specifically, we're currently working with this hypothesis that striatal dopamine might modulate cognitive control indirectly by changing the value of cognitive work. And according to, if you will, a modern gating model, the opponented actor learning model proposed by Anne Collins and Michael Frank, striatal dopamine increases the sensitivity to benefits over sensitivity to costs by increasing activity in go versus no-go pathways of the basal ganglia. And to test this hypothesis, we started to employ a task developed by Andrew Westbrook and Todd Braver, and we do this in collaboration with Andrew and Michael. And this is a cognitive effort discounting paradigm, where we can quantify the monetary value that people assign to performing a cognitive task, a difficult cognitive task. And this paradigm consists of 3 different phases. In the first phase, people are exposed to multiple levels of difficulty of a cognitive task, in this case an n-back task, so a 1-back, a 2-back, a 3-back, and a 4-back task. And in this initial training phase, they learn to associate each of these tasks with a unique shape. In the second phase, which was administered after drug administration, by the way, subjects choose between these shapes to indicate whether they would like to repeat these difficult or easy cognitive tasks after the experimental session, in return for a certain amount of monetary payoff. Now, critically, the monetary payoff for the easy task of the pair is titrated until equivalence is reached, a point of indifference is reached. And at this point of indifference, we can take the monetary amount of the easy task and use that as the subjective value of the difficult task. And that subjective value, that indifference point, is the key dependent measure here in this study. To assess the effects of striatal dopamine, we conducted a huge multi-session pharmacological fMRI PET study in 100 subjects. And this was team effort, as you can imagine. And as part of this study, these 100 subjects underwent an F-DOPA PET scan with the radio tracer F-DOPA, which indexes the degree to which uptake of which indexes the degree to which dopamine is synthesized in the striatal terminals of midbrain dopamine neurons. And it turns out there's quite a bit of variability, even in the healthy population, with some people exhibiting high dopamine synthesis capacity and others exhibiting low dopamine synthesis capacity. And interestingly, that dopamine synthesis capacity predicted the subjective value assigned to a cognitive task, that key-dependent measure plotted here on the y-axis, separately for the green high dopamine subjects and the blue low dopamine subjects. You can also see that, as predicted, the subjective value assigned to performing a task is much lower for a difficult task than for an easy task, which is plotted on the x-axis. Now, critically, this value of cognitive work is also modulated by dopaminergic drug administration, both of a non-selective catecholamine enhancer methylphenidate on the left, as well as by a selective dopamine D2 receptor agent, sulpiride, on the right. However, these effects were baseline dependent and seen only in subjects with low baseline levels of dopamine synthesis capacity. Subjects with high baseline levels of dopamine, in fact, exhibited, if anything, a reduction in the value of cognitive work. Now, interestingly, this opponent-actor learning model allows us to make more specific predictions about the effects of striatal dopamine. And specifically, this model predicts that dopamine should increase the weight on the benefits, but decrease the weight on the costs. Now, in a third phase of the experiment, Andrew sampled choices on either end of the point of indifference of each individual. And this allowed him to plot the full psychometric curve, which quantifies, in short, the degree to which people choose the high-effort task as a function of the difference between the subjective value of the high-effort task and the subjective value of the low-effort task. As predicted, see this clear S-curve. Now, intriguingly, as confirmed by OPAL model simulations, increases in the benefit are associated with a steepening of the slope on the right-hand curve, whereas decreases in the cost of work is associated with a flattening of the curve on the left-hand side. And it turns out that striatal dopamine has exactly this asymmetric effect on the psychometric curve as predicted by the model. So we see that individual differences in striatal dopamine, but also these dopamine drugs have these opposite effects on the slopes on the right and the left-hand side of this psychometric curve, implicating that it increased the weight on the benefits, but decreased the weight on the costs of cognitive work. So this then led us to conclude that striatal dopamine gates cognitive task in proportion to its benefits versus costs. And this then together serves to illustrate this general take-home message that chemical neuromodulators adapt structurally fixed neural systems to our changing environment. And dopamine might do so by allowing us to arbitrate or make decisions between different cognitive tasks or different cognitive strategies based on their costs and benefits. Of course, this question raises another key question, which we would like to address in future work. If the midbrain arbitrates between different cognitive strategies, different cognitive tasks, then what controls the midbrain? For dopamine, we can ask, how do we set the value of cognitive work? This is not a trivial question because valuation is now recognized to be context dependent. To illustrate that, cognitive work is not so valuable when the environment is uncontrollable, or when it is volatile, or when it is opportunity costly. So an intriguing hypothesis is that the dopaminergic midbrain is controlled by top-down signals from medial frontal regions that we know compute key statistical meta-parameters of the environment, like controllability, like its volatility, like its opportunity cost. More speculatively, we may ask whether the medial frontal cortex prioritizes prefrontal versus striatal midbrain projections by setting their respective baseline levels of dopamine. And indeed, we know from work, for example, from Antonio Strafella, who has combined TMS with dopamine PET, that transcranial stimulation of the cortex can change dopamine release. So this then leads us to the current working hypothesis for future work, which is that dopaminergic drugs might alter the balance between distinct cognitive strategies by having paradoxical effects depending on environment-specific baseline levels of dopamine in distinct neural systems. To go back to our example of Parkinson's disease, where the drug biased the system towards being more flexible but less stable, perhaps this reflects a remediation of severely depleted dopamine levels in the striatum, but a detrimental overdosing of dopamine levels in the prefrontal cortex. By contrast, in ADHD, the diametrically opposite effect of similar dopaminergic drugs might reflect a restoration of dopamine levels in the deficient cortex and an overdosing of levels in the striatum. And I want to make one final point, and that is that I would argue, and I have reason to do so, that these meta-level arbitration effects of dopaminergic drugs are unlikely to be restricted to this specific flexibility-stability trade-off, and are rather likely to extend to other computational trade-offs, such as, for example, the one between controlled processing and automatic processing. For example, what we've seen in 2 recent studies is that methylphenidate can restore a balance between an instrumental control strategy, often associated with the dorsal striatum, and a Pavlovian autopilot strategy, more often associated with the ventral striatum. And this restoration happened selectively in people with low mental capacity. It is those people with low mental capacity who exhibited very high autopilot effects at baseline, who exhibited drug-induced increase in an instrumental control strategy. Subjects who have quite high mental capacity to start off with exhibited, if anything, the opposite effect. The general point I want to end with is that the chemical neuromodulators are well suited to regulate key computational trade-offs. Their effects depend on the baseline tone of different projection systems, which might well reflect the perceived statistics of the environment. And therapeutic drugs counteract this bias elicited by the environment by restoring systems with low baseline tone and overdosing systems with high baseline tone. Of course, this raises multiple big open questions, including, might disordered behavior in mental illness represent perhaps a normative adaptation to the perceived statistics of the environment? And who then flips the switch? Can we manipulate the cortex, say with TMS, to alter top-down environment-specific control of the midbrain? And what is the timescale at which the midbrain switch can act? Can the switch be flipped according to state changes rather than trait differences? And with that, I'd like to end and just tell you to watch this website for the answers to this question in the next few years. Thank you very much.